Nobel Laureates, Excellencies, ladies and gentlemen, it's a great pleasure and indeed a privilege to welcome all of you to Karolinska Institutet's Aula Medica. We are here for the 2019 Nobel Lectures in Physiology or Medicine. My name is Ole Ottersen and I'm the president of Karolinska Institutet. During the Nobel Week, and particularly, I should say, during the Nobel Lectures, we celebrate science and rejoice at unique contributions of some exceptional individuals. Research is a way of quenching of a thirst for knowledge, testing our own limits, and having an outlet for our creativity and competitive spirit. I'm convinced we will be witness to all these facets of research as we attend the lectures here today. Within physiology or medicine, the ultimate goal is to achieve scientific breakthroughs that change or review of human health, disease, and normal vital processes, the fundamentals of life. Dear laureates, through your seminal discoveries, you have indeed made historic contributions to these areas. Today, it's an honor for me to extend my warmest welcome on behalf of Karolinska Institutet to William Kaelin Jr., Sir Peter Ratcliffe and Gregg Semenza, who will be receiving the 2019 Nobel Prize in Physiology or Medicine for their discoveries of how cells sense and adapt to oxygen availability. Without further ado, I will now call upon Patrick Ernfors, Professor of Molecular Neurodevelopment and member of the Nobel Committee for Physiology or Medicine. Professor Ernfors will introduce the laureates. Patrick, the floor is yours.
Ladies and gentlemen, we are at the most exciting point of the Nobel Week. We will hear directly from the laureates about the research that has been awarded this year's Nobel Prize. On behalf of the Nobel Assembly at Karolinska Institutet, it's a great pleasure for me to introduce to you the 2019 Nobel Laureates in Physiology or Medicine. Professor William Kaelin, Professor Sir Peter Ratcliffe, and Professor Gregg Semenza. They have received this year's prize in physiology or medicine for the discoveries on how cells sense and adapt to oxygen availability. The need for oxygen to sustain life has been understood since the onset of modern biology. Oxygen is used in oxidative reactions to convert food into energy useful for cells. But animals can be exposed to low oxygen levels. They can also vary between tissues and cells. And sensing and adapting to these changes is critical for survival. And because of this, during evolution, mechanisms have evolved that allow cells to adapt to changes in oxygen levels. But the mechanism by which organisms sense and adapt to oxygen has remained unknown. The story on this discovery has many surprises and starts with two apparently different lines of research. Studies on the regulation of the erythropoietin gene and studies on the function on the von Hippel-Lindau tumor suppressor gene in cancer. Working in these areas of research, Dr. Kaelin, Dr. Ratcliffe, and Dr. Semenza discovered a master regulator for cellular and systemic oxygen homeostasis. And they showed that this machinery exists in cells and tissues throughout our body, constantly monitoring oxygen levels and, when necessary, setting into motion processes which make cells and tissues adapt. The molecular details of oxygen response regulation did not stop unfolding until once the discoveries that were awarded with the 2019 Nobel Prize were made. On the contrary, the key discoveries of this year's laureates opened the field and led to an explosion of work that has uncovered further molecular complexity in response to oxygen flux. The oxygen-activated signaling pathway controls at least 300 genes, affecting a wide variety of functional properties, ranging from rewiring metabolism, immune and inflammatory responses, respiration, the generation of new blood vessels, altitude adaptation, and production of red blood cells. Since the seminal discoveries by this year's laureates, oxygen sensing has also developed into a research field with clinical importance. As expected, based on the fundamental importance of oxygen sensing in normal physiology, it is also central to major diseases. For example, anemia, ischemic vascular diseases, and cancer. And intense ongoing efforts now focus on developing drugs that target the oxygen sensing machinery. In addition to this year's Nobel Prize in Physiology or Medicine, the laureates have been awarded numerous prizes along their distinguished careers, including the Canada Gardner International Award, Scientific Grand Prix, Wiley Prize in Biomedical Sciences, Albert Lasker Basic Medical Research Award, and Mastery Prize. It is time to hear the laureates. We'll hear 3 individual lectures, and this is a unique opportunity to gain first-hand insights into the journeys that have led to their seminal discoveries, which explain how cells sense and adapt to oxygen availability. Our first speaker is Gregg Semenza. He is a professor of pediatrics and genetic medicine and serves as the director for the vascular program at the Institute for Cell Engineering at the Johns Hopkins University School of Medicine in Baltimore. He is a medical doctor and a geneticist, and a member of the National Academy of Sciences. Dr. Semenza received his bachelor degree from Harvard University in 1978 with the intention of pursuing a career in basic research in the area of genetics. For various reasons, he changed his mind and instead applied for an MD-PhD program and joined the University of Pennsylvania, where he earned his medical doctorate PhD degree in 1984. His PhD work involved the sequencing of the beta-globin gene of a family with beta thalassemia. Dr. Semenza completed his medical training in 1986 with an internship and residency in pediatrics at Duke University in North Carolina. He thereafter moved to Johns Hopkins University in Baltimore, where he has been ever since. He started there by attending medical genetics patients and working in the pediatric emergency room, and he also obtained a postdoctoral fellowship to study beta thalassemia. His idea was to study human genetic disorders by making transgenic mice carrying the human mutations. But by chance, he obtained a clone for the erythropoietin gene, and this would entirely change the course of his career. Together with John Gerhardt and Stylianos Antonarakis at the Johns Hopkins, he made a transgenic mouse that expressed human erythropoietin. With the help of his mentors, in 1989, when still a postdoc and without a faculty position, Dr. Semenza obtained 2 grants, one from the National Institute of Health, to study the regulation of the erythropoietin gene. With funding secured, he would, through tedious biochemical strategies, identify the proteins that bind to the regulatory region of the erythropoietin gene and he eventually managed to purify and clone these factors and named them hypoxia-inducible factors because of the presence of protein under low oxygen but absence under normoxia. This discovery opened the door to research on oxygen sensing. And Dr. Semenza has since continued his research, which ranged from basic science of the molecular mechanism of oxygen sensing to its medical implications. I would like to ask you all to join me in welcoming Professor Gregg Semenza to the stage, where he will deliver his 2019 Nobel Prize lecture.
Thank you for that kind introduction, and thank you for this tremendous honor to speak to you all about our research over the last 3 decades, really, trying to understand how the human body can adapt to changes in oxygen availability. And this is really a critical physiological property of every cell of the human body, because we know that cells must receive oxygen on a continuous basis. So every one of the 100 trillion cells in your body requires oxygen in order to make energy. And so this oxygen demand must be met by the oxygen supply and the delivery of blood, which carries oxygen through the blood vessels across the body to ensure that every cell receives adequate oxygen at every moment of the day. And the hypoxia-inducible factors serve this role to ensure this balance between oxygen supply and demand. So I'd like to tell 3 stories that illustrate how the hypoxia-inducible factors are important in both physiology and medicine. So there wasn't any need for the or this year. And I'll start where we started. As you heard from Professor Ehrenfors, we initially tried to understand the control of red blood cell production. And at the time that we started this work, we knew that red blood cells, of course, are produced in the bone marrow, but the signal that controls the production of red blood cells was made in the kidney. And it was known that individuals who had chronic kidney disease stopped making EPO and became anemic. And so it was a great breakthrough when the human erythropoietin gene was cloned, because it allowed the production of EPO in the laboratory, and that became useful as a therapeutic for the treatment of anemia in the context of chronic kidney disease. But what wasn't known was what controlled erythropoietin production, and in particular, what allowed cells in the kidney to produce EPO in response to changes in oxygen availability. And what we were able to do was to first identify a short sequence downstream from the human EPO gene that became known as the hypoxia response element. And this is a very short sequence consisting of 33 base pairs of DNA that has the property that it can be taken out of the human EPO gene, inserted into a foreign gene, and the expression of that gene will be induced by hypoxia, by low oxygen. And then we used this sequence to identify a protein that bound to it under hypoxic conditions, and we called this hypoxia-inducible factor 1, or HIF1. And we purified the protein based on its binding to the sequence and found that it was a heterodimer composed of 2 subunits that we called HIF1 alpha and HIF1 beta. And under hypoxic conditions, these two proteins dimerize. They bind to the hypoxia response element and recruit proteins called coactivators that serve as a bridge to the RNA polymerase that's responsible for transcribing the DNA into RNA. The important question that arose was, of course, what controlled the levels of HIF-1 protein under hypoxic and normoxic conditions. And as you'll hear from Peter Ratcliffe and Bill Kaelin shortly, this involved a novel post-translational modification in which two proline residues in HIF-1 alpha were hydroxylated under normoxic conditions. So when oxygen was available, an oxygen atom was inserted into HIF-1 alpha subunit, and this hydroxylation allowed the protein to be bound by the von Hippel-Lindau or VHL protein. And when VHL binds to HIF-1 alpha, it targets the protein for destruction. So when oxygen is available, the protein is hydroxylated and destroyed. However, under hypoxic conditions, when oxygen levels are decreased, the hydroxylation reaction is inhibited, VHL cannot bind, and the HIF-1 alpha protein rapidly accumulates within hypoxic cells. In addition to this hydroxylation of proline residues, there's an asparagine residue in the transactivation domain of HIF-1 alpha that's also hydroxylated by another protein that we identified and called factor inhibiting HIF-1 or FIH1, because this protein inhibited the ability of HIF-1 alpha to recruit the coactivator proteins. And it was subsequently shown that FIH1, like the Prolyl hydroxylases, was capable of inserting an oxygen atom into an amino acid residue of HIF-1 alpha, in this case an asparagine residue. And in this case, the hydroxylation prevents the interaction with the coactivators. Again, under hypoxic conditions, the hydroxylation does not occur. The coactivators can bind, and HIF-1 is competent to activate gene transcription. So through these oxygen-mediated modifications, the activity of HIF-1 is regulated, and this provides a direct way to transduce changes in oxygen availability to the nucleus as changes in the activity of HIF-1. Early on, after we had purified the protein and isolated the cDNA sequences, we generated antibodies against the HIF-1 alpha and HIF-1 beta proteins, and we used these antibodies to determine the levels of the proteins in cells as a function of the oxygen concentration that they were exposed to. And what we found is that oxygen concentrations below 6%, there was a dramatic increase in the HIF-1 alpha levels. This increase occurred in a graded fashion, so the more severe the hypoxia, the greater the levels of HIF-1 alpha protein. And 6% oxygen corresponds to venous PO2, and most cells in the body are exposed to between 3 and 6% oxygen. So any decrease in oxygen availability in the body occurs along the steep portion of this curve, providing a graded transcriptional response to hypoxia. And we started out by identifying the binding of HIF-1 to a hypoxia response element in the EPO gene. But now we know of over 4,000 genes that are regulated in a similar manner by the binding of HIFs to sequences within the gene that result in activation of gene expression when oxygen levels are reduced. And of course, hypoxia can result either from an increase in the amount of oxygen that's consumed. So, for example, if there are more cells consuming more oxygen, as in a tumor, or a decrease in oxygen delivery. So, for example, in the context of coronary artery disease, where the blood flow to part of the heart muscle is impaired, this will result in the induction of HIF-1 and the expression of downstream target genes. And we know that, for example, in the case of the EPO gene, this is a response to systemic hypoxia. So if we go to high altitude, every cell in the body will be exposed to hypoxia, and the response is systemic, an increase in red blood cell production to increase delivery of oxygen to all the cells. However, in the case of coronary artery disease, there's only a small area of the heart muscle that's deprived of oxygen, and so the response is a local response. For example, the production of vascular endothelial growth factor and other proteins that stimulate blood vessel growth in order to increase oxygen supply to the hypoxic tissue. And then regardless of whether the hypoxia is systemic or local, individual cells have to adapt their metabolism to changes in oxygen. And the really classic metabolic adaptation to hypoxia is switching from respiration, which uses oxygen, to glycolysis, which allows energy production in the absence of oxygen. And this, of course, helps cells to survive oxygen deprivation. Early on, we were able to knock out the expression of the HIF-1 alpha gene in mice, and we showed that mice that were homozygous for the knockout allele, so these animals did not make any HIF-1 alpha protein, the development of the mouse embryos arrested at mid-gestation, and there were defects in the development of the heart, the blood vessels, and the production of red blood cells. So all three components of the circulatory system are dependent on HIF-1 for their normal development. Now, of course, because these embryos did not survive to be born, we could not study the effects of complete HIF-1 deficiency in adult animals. But what we found was that animals that were heterozygous, that contained one of the mutant genes and one of the wild-type genes, developed normally, but as you'll see, had impaired responses to hypoxia and ischemia. Well, it turned out that in addition to HIF-1 alpha, there were several siblings hiding in the human genome. One of these was named HIF-2 alpha and the other HIF-3 alpha. And these three proteins are products of different genes, but they have related protein sequences. The proteins are all stabilized under hypoxic conditions through the mechanisms that I described, and all three can heterodimerize with HIF-1 beta. And whereas HIF-1 alpha is present in all nucleated cells of all animal species, HIF-2 alpha and HIF-3 alpha occurred only during vertebrate evolution. And these are only expressed in certain cells during the development of vertebrates such as humans. So we had done these experiments in animals, but what was the proof that this system was important for red blood production in humans? And of course, the answer is genetics. And there were patients identified who made too many red blood cells. And these patients were prone to having strokes and other thrombotic disorders, and we and others attempted to identify the mutations that were responsible for this phenotype called congenital polycythemia. And the first mutations were found in the receptor for EPO that's present on red blood cell progenitors. And this mutation makes the receptor hyperactive so that it stimulates red blood cell production even in the absence of the EPO protein. The second group of mutations that were identified were in the VHL gene. And in the VHL gene, a change of a single amino acid in the protein altered the ability of the VHL protein to interact with the HIF-1 alpha and HIF-2 alpha after they had been hydroxylated. And so as a result, less of the HIF proteins are degraded at any oxygen concentration, so there's more HIF activity, and this results in increased red blood cell production. Other individuals were found not to have mutations in either of those genes and turned out to have mutations in one of the Prolyl hydroxylase genes. And these mutations, again, a single amino acid change in the protein, decrease the ability of the enzyme to insert the oxygen atom into HIF-1 alpha and HIF-2 alpha. And then finally, families were identified where there were mutations in the HIF-2 alpha gene itself. Again, changing only one amino acid in the protein, and these amino acids made it more difficult for the hydroxylases to bind and modify the protein. So you can see that this pathway is virtually saturated with mutations, demonstrating the critical role for this physiological process in human beings. And it's somewhat ironic, of course, that where we started with the EPO gene and HIF-1 alpha, these are the only components in the pathway that have not been shown to have mutations in this disease. So these are the kind of ironic findings that bring us great joy, actually, because they're unpredictable. So now I'd like to turn to disease conditions and first illustrate the role of hypoxia-inducible factors as protection against human disease. And I'll start with ischemic cardiovascular disease, which, of course, leading cause of death in the industrialized world. And, of course, this comes in two major types. If there's narrowing of a major coronary artery, this, of course, can result in angina or chest pain, and that can eventually lead to myocardial infarction, a heart attack. And then if the arterial obstruction occurs in a large blood vessel in the leg, this results in leg pain and can eventually result in a condition like this, where you can see that there's an obstruction to blood flow here, and the only blood, this is about the level of the knee here. So here's the blood vessel in the thigh, and then you can see below the knee there's very little blood flowing to the lower leg, and the only blood flow is coming from these blood vessels that branch off above the obstruction. And this is a condition that's known as critical limb ischemia. It's critical because the amount of blood flow that's reaching the lower leg is not sufficient to maintain the viability of the tissue. And so these patients will have ischemic pain at rest. They'll develop ulcers that will not heal. Eventually, gangrene will occur. And in many patients who are not able to be rescued by some sort of revascularization, this will require limb amputation. It's quite hard to believe this, but there are thousands of patients who have their limb amputated every day because of adequate therapy for this condition. So we were interested in modeling this disease in mice, and we were particularly interested in the fact that the peripheral arterial disease is really an age-related disease. So the older patients are much more likely to develop critical limb ischemia. And so we looked at the effect of aging in a mouse model where we were able to ligate or cut off the major artery that supplies blood to one of the legs. And we used a technique called laser Doppler perfusion imaging in order to look at the blood flow non-invasively. And you can see that before surgery, the ratio of blood flow into two limbs is equal. So the ratio is 1. And then after the surgery, there's a great decrease in blood flow on this side where the operation has been performed. And so the ratio drops very low. And then over time, in young animals, you can see that there's this dramatic recovery of blood flow. However, if we look at older mice, the recovery of blood flow is not quite as good. And then in very old mice, because mice only live to the age of 2 or 2.5, there's very little recovery of blood flow at all. Then at every age, we compared animals that had a mutation that knocked out the expression of HIF-1 alpha in one of the two alleles, and these are called heterozygous HIF-1 alpha null mice, or het mice for short. And you can see at every age, the het mice recover less well than their wild-type littermates. So just by changing two parameters, the age of the mice and the genotype at one locus, we can cause this range of outcomes from very good recovery of blood flow to very little recovery at all. And this has important clinical consequences, so we can examine the mice and look for tissue injury. And in the young wild-type animals, there's 100% limb salvage, meaning there's no permanent tissue injury. But if we look at the 8-month-old mice, 40% of these animals had either death of tissue of the skin and soft tissue, or the loss of several toes, below the ankle amputation. And then if we look at the 20-month-old animals, you can see that about 15% of the animals had above the ankle amputation, meaning they lost the whole foot because of the inability to recover sufficient blood flow. And you can see at every age, the heterozygous mice have more severe phenotype compared to the wild-type littermates. Well, we looked at the levels of HIF-1 alpha protein in the ischemic compared to the non-ischemic limb 3 days after femoral artery ligation, and you can see in the young wild-type animals there appears a very strong production of HIF-1 alpha protein. In the 8-month-old animals, you can see the production is not quite as brisk, and in 20-month-old animals, there's very little induction of HIF-1 alpha at all. And you can see that the heterozygous knockout mice are also impaired in their production of HIF-1 alpha protein. So we see a correlation between the effects of aging on the induction of HIF-1 alpha protein and the recovery of blood flow, suggesting that one of the major effects of aging is to impair this normal physiological response to hypoxia and ischemia. If that were true, then we hope that we could provide replacement therapy through gene therapy. And so we engineered an adenovirus that encoded a modified form of HIF-1 alpha with a deletion and several point mutations that rendered the protein stable, so it would no longer be degraded under normoxic conditions. And so at the time that we performed the femoral artery ligation, we injected the adenovirus into the leg along the course of the artery that we had removed, and this was either this what we called a constitutively active form of HIF-1 alpha, or an adenovirus that encoded a bacterial enzyme. And you can see that the animals that were treated with the control virus recovered about 55% of their blood flow in 3 weeks, and the animals that were treated with the HIF-1 alpha adenovirus had a much better recovery of blood flow. Now, this was done in the young animals, and as you recall, the young animals can recover well on their own. So the question is, what happens when older animals are treated? And here you can see that the control animals, again, have a much less brisk recovery of blood flow. So compared to the recovery here, it's dramatically impaired in the control mice. And when we give the HIF-1 alpha adenoviral treatment, you can see that the level of recovery in the treated older animals is now similar to the recovery of the control younger animals. So we've been able to overcome this age-related impairment of vascularization through HIF-1 alpha gene therapy. And so we hope that it may be possible to engineer therapies in patients where we can provide the replacement therapy that will allow these patients to avoid limb amputation. And the reason that we think that this is particularly powerful is that HIF-1 does not just control the expression of vascular endothelial growth factor, but it is a master regulator that controls the expression of a whole series of proteins that are made by the ischemic cells, are secreted into the bloodstream, and then bind to receptors on endothelial cells in blood vessels, endothelial progenitor cells, mesenchymal stem cells, and other bone marrow-derived angiogenic cells that together are orchestrating an increase in tissue vascularization to improve blood flow and oxygenation. And again, what we found was that under the conditions of aging, that the expression of all of these enzymes, all of these proteins was impaired. And then when we performed the HIF-1 alpha gene therapy, we saw an increase in the expression of all of these genes. Well, as I've described, the role of HIF-1 is a physiological regulator of red blood cell production and vascularization. And now I'd like to tell you a little bit about the dark side of HIF-1, which is cancer. This is a typical picture of an advanced human cancer. These white areas are areas of necrosis where the cancer cells have died. And they've died because they're too far away from a blood vessel, because all the cells between them and the blood vessel have consumed all of the oxygen. And so these cells have died, but you'll notice that there are cells immediately around the areas of necrosis that stain very intensely with this brown stain. And that's because we've applied an antibody that binds to HIF-1 alpha protein. So these cells are making incredibly high levels of HIF-1 alpha protein in the nucleus. And we believe that these cells play a particularly important role in the invasion and metastasis of cancer and the ability of cancers to avoid killing by our therapies and also by the immune system. And whereas most of the cancer chemotherapies target rapidly dividing cancer cells that are present around blood vessels, there are no approved therapies that target these hypoxic cancer cells that express very high levels of HIF-1 and HIF-2. And so this immunohistochemistry using antibodies against HIF-1 alpha on primary patient biopsy samples have shown in a large variety of human cancers that high HIF-1 alpha levels are associated with increased patient mortality. So looking at the biopsy at the beginning of the patient's treatment, one can predict patients who are at high risk of failing therapy and dying of the disease by their high expression of HIF-1 alpha. And I've highlighted 5 out of actually 10 studies or so in breast cancer associating high HIF-1 alpha with increased patient mortality. So we were interested in identifying drugs that inhibit HIF-1 that might be used, first of all, in our cancer models in animals as a way of evaluating whether HIF-1 inhibition might be useful for cancer therapy. And so we identified several drugs that inhibit HIF-1 by different mechanisms. And the first one I'd like to describe is acroflavin. And this was one of Ehrlich's original antimicrobials. And it turns out that acroflavin can bind to HIF-1 alpha and HIF-2 alpha and prevent their dimerization with HIF-1 beta. And without dimerization, the protein is inactive. So we showed that acroflavin would inhibit the production of different HIF target genes. And when we treated tumor-bearing mice, what we saw was, so in this case, we've taken prostate cancer cells, injected them into an immunodeficient mouse, and waited for the tumor to grow, and then started treatment either with saline or with acroflavin at different concentrations. And you can see that the animals treated with saline injections, the tumor grows very rapidly, whereas the animals treated with the HIF inhibitor, there's very little tumor growth after we start the treatment. After 2 weeks of treatment, we removed the tumors and analyzed the expression of different genes that are regulated by the HIFs. And you can see that compared to the controls, there's a marked decrease in the expression of every HIF target gene that we looked at. And then if we take sections of the tumor and investigate the presence of blood vessels, this brown staining indicates blood vessels, and it's very easy for you to see them in the tumors from the control-treated animals, whereas in the animals treated with the HIF inhibitor, it's very hard to see the blood vessels because they're much smaller. and therefore, of course, there's much less blood flow, and as a result, the growth of the tumor is impaired. We've been interested in particular in breast cancer, and this is a model of so-called triple-negative breast cancer. These are breast cancers that do not respond to treatments targeting the estrogen receptor, the progesterone receptor, or HER2, and as a result, they're treated with cytotoxic chemotherapy, and the women often have a relapse after treatment and account for much of the mortality of breast cancer. And what we found was that the expression of the HIFs was particularly high in these tumors. So we injected one of these tumors into mice, into the mammary gland of the mouse, and these tumors will grow rapidly in the mice if we treat only with a saline control. If we treat with a HIF inhibitor, in this case a drug called digoxin that blocks the production of HIF-1 alpha protein, we see that the primary tumor growth is impaired. Compared to the control, the metastasis of cells from the primary tumor to lymph nodes is greatly decreased. And then finally, the metastasis through the blood to the lungs. These purple areas are all areas of breast cancer cells that have metastasized to the lung in the saline-treated animals. And you can see in the animals treated with the HIF inhibitor digoxin, there's no metastatic cells in the lungs. So we also looked to see what would be the effect of combining a HIF inhibitor with cytotoxic chemotherapy, in this case a drug called gemcitabine. And you can see that when gemcitabine is administered to the animals, the tumor growth is greatly impaired, but as soon as we stop treatment, the tumor growth resumes at the same rate as in the control animals. Whereas if we treat with gemcitabine and the HIF inhibitor, we can eradicate the tumor in this animal model. We've gone on to look at different steps in the metastatic process, so we can break down this into individual steps and then identify specific genes that are regulated by the HIFs at each of these steps, and show that the expression of all of these genes is inhibited when we administer one of these drugs. We were particularly interested in looking at the regulation of the breast cancer stem cells. So most of the cells in the tumor divide very rapidly, but can only divide a certain number of times and then stop dividing. But the cancer stem cells have the property of unlimited division. So eradicating these cells is critical for eradicating the tumor. And what we found is that when cancer cells were exposed either to hypoxia or to chemotherapy, there was a great increase in the expression of proteins that control the breast cancer stem cell phenotype. And these are proteins, NANOG being one of them, that were first identified in embryonic stem cells and are responsible for the stem cell phenotype in ES cells. And what we found was that all of these boxed symbols represent genes that are induced by either hypoxia or treatment with chemotherapy and are controlled by the HIFs. And they all lead through different pathways to the production of this protein NANOG that determines the cancer stem cell fate. If we treat with chemotherapy and a HIF inhibitor, we don't see any increase in NANOG production. Finally, we've been interested in the role of HIFs in controlling the production of proteins that allow cancer cells to avoid killing by the immune system. And these come in two types. The first is what's called the adaptive immune system, and these are particularly T lymphocytes that can recognize cancer cells as foreign and kill them. But cancer cells express several proteins. One of them is a protein called PD-L1 that binds to a protein called PD-1 on T cells and causes the T cells to become anergic or can even kill the T cells. A second protein called CD73 makes a small molecule called adenosine that binds to a receptor on T cells and has the same effect. It causes the T cells to become inactive or to actually kill them. Then a second branch of the immune system that can kill cancer cells is called the innate immune system, and this consists principally of macrophages, and macrophages are very good at eating cancer cells. So the macrophage will wrap itself around the cancer cell and eat it. However, if the cancer cell expresses a protein called CD47, it signals to the cancer cell and it blocks the ability of the macrophage to kill the cancer cell. So you can see that all of these proteins are induced when cancer cells are exposed to either hypoxia or chemotherapy, and we think that that this is an important mechanism by which these cells can evade killing by the immune system. And again, when we use HIF inhibitors, we find that the expression of these genes is blocked, and we can prevent the loss of active T cells within the tumor microenvironment. So we're quite interested in developing inhibitors that could be used in patients. The drugs that I've described to you are good for our animal models, but for a variety of reasons are not appropriate for use in patients. And so a great effort in my lab now is identifying new inhibitors that will block the HIFs and will be appropriate for use in patients. I just like to summarize then what I've tried to illustrate to you, the role of the HIF pathway in biology and medicine through development, physiology, disease, and even in evolution, where people who live at high altitude in Tibet and the Andes have genetic alterations in the HIF pathway that allow them to adapt to life at high altitude. Finally, I'd like to dedicate the lecture to Dr. Rose Nelson, the late Dr. Rose Nelson. She was my high school biology teacher and my inspiration in science, and I'm here because of her. The other people I would like to thank are my mentors, Haig Kazazian and Stylianos Antonarakis, who are here today. They were very generous in helping me and allowing me to have independence to pursue this project. And I can't thank them enough. And the late Victor McCusick, who also was extremely generous in helping me in my early career. I'd also like to acknowledge two faculty members at Hopkins, John Gearhart and Tom Kelly, who played critical roles in our studies of the erythropoietin gene and the purification of HIF1, and Guang Wang, who was my first postdoc and who purified HIF1. Finally, colleagues that I've collaborated over the years, Larissa Shimoda, Chidong, Akrit Sodhi at Hopkins, Joe Prakal at the University of Utah, Nanduri Prabhakar at the University of Chicago, and finally Mike Armstrong, who had endowed a professorship that I hold and has provided us with great support in our cancer research. And then finally, I'd just like to illustrate that this work was done by my lab, not by me, and that I've been very fortunate to have a large group of undergraduate students, graduate students, and postdocs over the years who have performed these studies. I've been fortunate to have many colleagues on the Hopkins faculty with whom I've collaborated, and then faculty elsewhere around the world that we've worked with to try to understand the role of HIFs in physiology and medicine. And last but not least, I'd like to thank, Thank you. Okay, I've got it together. I'd like to thank my wife, Laura, my sons, Evan and Gabe, my daughter, Allie, for always being there for me, giving me unconditional love and support. Thank you so much.
Professor Sir Peter Ratcliffe is the Director of Clinical Research at the Francis Crick Institute in London and the Director of the Target Discovery Institute at University of Oxford. He's a medical doctor and trained as nephrologist. He is a Fellow of the Royal Society in London. Dr. Ratcliffe studied medicine at the University of Cambridge with clinical practice at Bartholomew's Hospital in London, and he obtained his medical degree in 1978. He worked at different postgraduate hospitals in London for a few years and thereafter relocated to Nuffield Department of Medicine at Oxford University to train in nephrology. In 1987, he presented his MD thesis that was focused around renal oxygenation. Dr. Ratcliffe was to stay at Nuffield Department of Medicine for many years, and in 2004 he became the Nuffield Professor and Head of Nuffield Department of Medicine, University of Oxford, and these positions he has held until 2016. Having mostly been involved in clinical studies and studies on renal physiology in rats, Dr. Ratcliffe obtained a senior fellowship from the Wellcome Trust in 1919 for research on an entirely new topic, oxygen sensing. And since then, Dr. Ratcliffe has continuously contributed in clinical research, renal physiology, and of course, most importantly, to the mechanism of oxygen sensing and its therapeutic opportunities. Among his vast contributions to science, Dr. Ratcliffe placed von Hippel-Lindau protein at the center stage of oxygen sensing, and he discovered the biochemical link between oxygen availability and oxygen sensing. I ask you all to join me in welcoming Professor Sir Peter Ratcliffe to the stage, where he will deliver his 2019 Nobel Prize lecture.
Thank you very much indeed, Patrick, for that kind introduction. Thank you also to the Nobel Assembly for this very great honour. I should like to begin this lecture, since Gregg, Bill and I are an Anglo-American trio, with the Anglo-American expedition to Pikes Peak, Colorado, to study altitude, that's hypoxia, adaptation, a little over 100 years ago. The expedition was led by John Scott Haldane on the left there, but the heroine for this lecture was surely Mabel Fitzgerald, the only female member of the expedition, not allowed to be a member of Oxford University on account of her gender. But here she is with the men at the summit of Pikes Peak, where there was a very comfortable hotel to base their laboratory. But the decision was that Fitzgerald should work elsewhere to make measurements at intermediate altitude in the surrounding mining towns. The story is that this is because the men were uncomfortable that she was unchaperoned in that comfortable hotel. But off she went on her own to visit those towns that were more or less the Wild West. And these are the measurements she made. on the top there of breathing and below of changes in the blood, in each case showing the very sensitive changes in each of those parameters to small changes in altitude and hence oxygen. This wasn't the first demonstration of adaptation, but I think it was the first demonstration of the extraordinary sensitivity of the hypoxia pathway that we're going to discuss this afternoon. The changes in the blood were, as Gregg has alluded to, mediated by the hormone erythropoietin. And here again, looking directly at EPO, we can see that extraordinary sensitivity here, just a donation of a unit of blood. EPO is made in the kidney. As you've heard, I'm a kidney specialist by training, and this was this extraordinary sensitivity that drew me to the subject to find out what was the oxygen sensor. Now, as Gregg indicated, there was a new opportunity for us at this time, since the EPO gene had been cloned. We were able, we thought, to go outwards from the EPO gene progressively to that sensor, and I'm going to describe my own lab's experience in that journey. We imagined that we would be moving out to an extremely specialized sensor in extremely specialized cells just within the kidney, which sense oxygen and made EPO. The only problem was that those cells were not identified in the kidney and were not available in a convenient tissue culture dish for our studies. So we tried to make them grow out of kidney by expressing an oncogene, T antigen, to make the cells proliferate and grow out of kidney. This experiment did identify the cells along with the work of Kai Eckhart. Here, the rather nice immunohistochemistry in my lab from Patrick Maxwell identified not as one of the interesting cells of the kidney, not obviously specialized, the rather humble fibroblasts, but still they wouldn't behave in growing tissue culture. So great credit should go to Franklin Bunn, who identified in some hepatoma cells, hepatoma from the liver, the other organ makes EPO, that made EPO in tissue culture, and importantly increased it very much in low oxygen. And those were the cells that we, and Gregg's alluded to the work in his lab, first used to try to map the control sequences at the erythropoietin gene as a first step in that journey, which we knew must interact with the upstream sensing mechanism. And this was our work on the mouse gene, identifying that 3' enhancer that could confer oxygen regulation on a reporter gene and hence must interact with the upstream apparatus. Chris Pugh and I worked night and day on these extremely cumbersome RNase protection assays that you see here that demonstrated that first step. But then I wanted to take a shortcut. I wanted to be clever and expression clone the upstream bits of the hypoxia signaling pathway by transferring genes from cells that had oxygen sensing, that was Frank's hepatoma cell, to one that didn't. That would be every other cell. And to my surprise and initial irritation, it was necessary to show this cell, the control cell, didn't have the property in the first place. And to my surprise and initial irritation, that was not the case. We found the control cell had the property also. Now, Patrick Maxwell took this work on, and here are some of the protection assays that Patrick did. And they show that every cell, in fact, that we tested, ultimately on the right conditions, had this property. And we realized that this was of pretty substantial significance, since it implied that extraordinarily sensitive system that had been shown first by Fitzgerald and then by those EPO would be was present in all cells, which must be doing other things. Since they don't make EPO, they must be regulating other genes in that way. So I was very excited by the work and wrote it up, and not one to take a chance. I drove with the manuscript to the offices of a very famous journal in London, and my aim was to burst in and see an editor, and I believe the editor would immediately see the extraordinary significance of this work, like myself, and agree to publish the work. This was Maunday Thursday, the day before Easter. Agree to publish it immediately after Easter. This is the letter we received about 4 months later on. And for the young people, if they ever say this to you, we had great difficulty finding reviewers. This is good. This means you're in quite a small field. It's going to make life a lot easier for you. Anyway, that was the first bit of my story. Gregg did the magnificent work that you've heard of identifying HIF that binds to that sequence. We met in the Lake Lucerne Palace Hotel shortly after this sadness, and he published for the first time, he presented for the first time unpublished work on his identification of HIF in those hepatoma cells, and I presented the work that I've just described to you. Here we are. At the eagerly contemplating the way forward. Do not be deceived by this photograph. The Lake Lucerne Palace Hotel was the most luxurious place either of us had ever seen before, and we're ever likely to see again until we came here. So anyway. We went back to Oxford, and of course the question was, what were the other genes? And this was our attempt on it. The phosphoglycerate kinase gene, a glycolysis implied in inhibition metabolism, and there is HIF binding the phosphoglycerate kinase promoter. So glycolysis, of course, is, some people will know, conserved throughout evolution. So we were keen to go back and look in more primitive species. Was HIF there too? And this was another piece of work identifying it in the fly cells, Drosophila, fruit fly, SL2 cells. You can see the rather clear HIF there. We still weren't making much impact with our editorial friends. This paper had the distinction of being first lost and then rejected by an editor of the Journal of Biology Chemistry. But the story in invertebrates is important, so I'll come to later on. As you heard very nicely from Gregg, the field exploded and whole new realms of understanding of hypoxia biology following these discoveries, and entirely new fields, and I've set them out just there. But at the time, we wanted to go upstream from HIF, and Gregg had importantly identified the encoding genes. So it was possible to ask what aspects of HIF conveyed oxygen sensing, and Gregg and others, fused bits of the HIF to an artificial transcription factor, what would confer oxygen sensitivity. We found these three regions would all do it. So each of them had to interact with this upstream process and then be transduced from the sensor to a piece of sequence. Now, everyone knew that protein phosphorylation was the means of signal transduction. So there had to be a phosphoacceptor amino acid in each of those regions to receive the signal. But Chris Pugh mutated every phosphoacceptor in this region. It made not a bit of difference at all. This work wasn't all that popular since it was entirely negative, but it did exclude the mainstream hypothesis. So what did those sequences interact with? It turns out there's the ones that interact with the von Hippel-Lindau gene, and well, this was a eureka moment in my lab. In VHL defective cells, HIF is stabilized, and if you put VHL back in, it gains that oxygen-sensitive property. But, you know, knowledge builds on knowledge, and this eureka moment didn't happen just out of the blue. Other people contributed importantly to the creation of that knowledge, importantly Bill here. But Gregg and I and others were finding these multiple transcripts you've heard about. Bill and others noticed that they were constitutively upregulated in these VHL defective cells from kidney cancer, and they were upregulated irrespective of oxygen. But puzzlingly, they didn't identify the mechanism. We heard about the work in Sheffield on a rainy day. It rains hard in Sheffield town in the north of England. We rushed back to Oxford, obviously to blot for HIF, and this is what we found. Very puzzling, and I presume it was puzzling in Bill's lab too. Another strand of work, Steve McKnight was looking for other pass proteins in the human genome. He found something that might have been a paralog of HIF, called it EPAS, endothelial pass protein. But it needed an antibody to demonstrate whether it's oxygen sensitive or not. Patrick Maxwell in the lab made one, PM9, Patrick Maxwell 9, and Patrick Maxwell 9 was responsible for this blot and the déroulement. Actually, these particular cells, many of them only express HIF2, and that's the demonstration why the blot was initially blank. So this experiment had quite a number of important outputs. Amongst other things, I won't have time for this, but why is the distortion to HIF2 in RCC? As I think Bill will allude to, HIF2 is now a target for RCC, but importantly for this lecture, it was one step further along the oxygen sensing pathway. VHL is a ubiquitin ligase, and in the presence of oxygen, it destroys HIF, and in the absence, HIF builds up. A lot of this work in my lab being Norma Masson and Matthew Cockman following on. So that gave us a biochemical question. What was the nature of that in that extract that caused these two proteins to bind together? And here we needed to correct a glitch, and this was the very important and beautiful work of Panu Yakala here and David Mole. We had noticed that the association was broken by cobalt, FO. These are compounds that mimic hypoxia, but apparently not by hypoxia itself. That was very puzzling to us. why not oxygen? Andrew Skin from Baker Ruskin visited the lab and sold me a hypoxia workstation. I had a bit of spare money from the Wellcome Trust, which is quite important. That's a plug to the scientists have spare money. And we bought that thing, and Panu and David were then able to reperform the immunoprecipitations with all the buffers equilibrated to hypoxia and perform and then we understood that the reaction was indeed oxygen dependent. So it had these characteristics of oxygen and iron dependent. It wasn't ATP dependent, consistent with the absence of any evidence for protein phosphorylation in the signaling pathway. We were always suspicious of a particular Prolyl residue, but this is one of the moments you live for. We made ultimately a peptide with the proline residue hydroxylated, and you see here with beautiful clarity that you no longer need to treat the extract then, the HIF then with extract binds to VHL very powerfully. In any case, this was the work of Yamin Tian. And it had a great significance because that was the evidence that likely the modification was hydroxylation of proline, and that immediately suggested an oxygen sensing mechanism through prolyl hydroxylases, which consume oxygen. And Bill will tell his side of that story. We a little bit more fortunate with the editors these days. We publish those papers back to back in Science. This was the position then that it was known that the collagen prolyl hydroxylases, oxygen splitting enzymes, they require iron and cobalt, which were those hypoxia mimetics. So we're in some ways rather good candidates anyway, but we couldn't believe the collagen prolyl hydroxylases could do it. Amongst other things, the sequence was wrong, and Johanna Mullaru from Finland was able to exclude that for us biochemically. So what were the enzymes was the next question, and knowledge builds on knowledge, so this is another curious one. We met up with this guy, Chris Schofield, who was to become a very valued long-term collaborator. Now, Chris was working on 2 oxyglutarate oxygenases, but not the collagen enzymes, quite a different thing. He was studying enzymes involved in antibiotic synthesis, and there's one of the first structures. That triad of iron coordinating residues was the signature that enabled him to predict other members of this family, including some that might be prolyl hydroxylases. Meantime, others have been working on mutant C. elegans worms, 145 catalogued mutated worms that couldn't lay eggs. That was the subject of this paper by Bob Horowitz, a previous recipient of this honour. Egg laying, well, a neural, neurosecretory, neuromuscular defect. What could that possibly have to do with what we were looking for? But they catalogued all those mutants. Now, earlier I told you we looked for HIF in flies. John O'Rourke in the lab also looked for it in worms, in C. elegans worms. And this time, critically, he made an antibody. And this is that antibody, rather beautifully showing regulation by oxygen in the worms. Andy Epstein took this project on, and he looked at all sorts of worms. You don't need a license, no expertise, poor worms. They all went in that bell jar, and all of them responded to hypoxia in the way that you see. But one of Chris's mutants, EGL9, look at that, completely constitutive HIF. I can remember Andy Edstein bursting through the office door saying, "Here's your gene." And another of those moments you live for, it sure was. One of Chris's candidates, Egl9, is the Prolyl hydroxylase that we're talking about, the oxygen sensor. So that's the system, simply a regulatory oxygen-splitting enzyme that adds an oxygen atom to the proline ring. The alcohol group here then hydrogen bonds to VHL and degrades HIF in the presence of oxygen. That's the switch that regulates all those responses you've heard about from Gregg. That's that Fitzgerald first noted in those Wild West mining towns in Colorado. Here she is again, both the acclimatized, increased breathing sensitivity to oxygen and the hemoglobin response, both the result of switching the HIF. And we can see here the carotid body that does those breathing responses, expressing HIF protein and lots of HIF mRNA, and those lowly fibroblasts in the kidney expressing HIF and doing the hemoglobin response. That work of other female scientists in the lab, Tammy Bishop, Emma Hodson, and Johanna Lima. So from EPO to oxygen, there we are. But we learned so much more along the way. All of these unexpected aspects of oxygen physiology, and that 2-oxyglutarate, the co-substrate, you make an analog of that, and it makes a drug that can be used for therapeutic manipulation of this system. I'll finish with that at the end. But in the last part of the talk, I want to talk about some new work. HIF, prolyl hydroxylase VHL, in that assembled form, appears only to be present in animals, in metazoan life. But it's since become clear that all 4 eukaryotic kingdoms apparently use this rather odd system of enzymatic protein oxidation coupled to proteases to signal oxygen levels, but in somewhat different ways. So here is the system in animal cells I've just described to you. Here is the system in protists, in dictyostelium, the very nice work of Chris West. It's similar but a little different in the ligase is the target of hydroxylation, not the substrate of the ligase. Here is the system in fish and yeast, the very nice work of Peter Espenschade. Here are different prolyl hydroxylase responding on a different transcription factor involved in the sterol response. But this is one I want to finish with, slightly different in plants. Plants, this is a very nice work of Francesco Lacozzi, who showed that plants use a different enzyme, cysteine oxidases, to dioxygenate an N-terminal cysteine in ethylene response factors, the sort of plant equivalent of HIF that enables the plant to make responses to things like flooding. If you overwater and kill your plants, you're probably killing them through hypoxia, and this is the way the plants try to adapt. If you overwater them, they inhibit the destruction of those transcription factors to bring on their own adaptive responses. So the work in our lab on this comes from a chance meeting with Francesco in Rome. Francesco asked me if he could express the animal system in plant cells as an exogenous indicator of hypoxia. I said, why don't we express the plant system in animal cells and see what happens? And this is the very nice work of Norma Masson, Tom Keeley, and Beatrice Guntoli, who came over from Francesco's lab. They fused that plant ethylene response factor sequences to a reporter, and to our surprise, when expressed in human cells, even without the plant oxygen sensing enzyme, it behaved in an oxygen sensitive way, and there you can see induction by hypoxia. The characteristics are different from HIF, but there it was. And when we mutated that target cysteine residue here, we saw, you can see there it doesn't respond, it's constitutive stabilized, as we've seen in some of the HIF system. So the implication was there's a human enzyme, the same as that plant enzyme, doing the same thing. And I hope you'll like this experiment. I love it as a human cell biologist and physician, conducted in Francesco's lab. These are the Arabidopsis seedlings, and this is one with the 4 of the 5 plant cysteine oxidases knocked out. It's looking rather poorly. We identified the human enzyme, and Francesco expressed that in the plants. And there we go, made the plants better, beautiful complementation. So ADO is the human ortholog of the plant oxygen sensor, another type of human oxygen sensor, and this is work in progress. We don't know how important that will be at the moment. We don't know whether there'll be medicine behind it, but it does have some rather interesting targets. The regulator of G protein, and you can see here that in the ADO knockout cell, that protein is stabilized. And when we look at G protein signaling in the ADO knockout cell, the regulator, the down regulator, is up, and the G protein signals are down. This the very nice work of Tom Keeley. So that's coming to the end now. I've and Gregg's shown you this amazing human oxygen sensor doing hundreds or thousands of things. I've drawn it in a very simplified way. We've alluded to the plant system, which is also very complicated but works in a different way. It has a reflection in animal cells. And then, interesting enough, what I didn't show you, HIF actually also interacts with those RGS proteins. And every time we look at HIF, we find another realm of complexity, seems to interact with the acute oxygen sensing system. So out of all that, how could you get the little bit you want to treat a human disease specifically? How can you get what you want? Think about that. But in fact, Bill will go through this, the results of giving those hydroxylase inhibitors in kidney patients are pretty promising. There's newer data from this. So far, these inhibitors show powerful effects on renal anemia with little evidence of general activation against that expectation. You can't always get what you want, but if you try, sometimes you find you get what you need. The words of Sir Michael Jagger and Mr. Keith Richards, of course, by repute, they knew a bit about drug discovery. And what they meant was that there was an element of empiricism, of trial and error in obtaining drugs. Molecular biology, we can take it so far, deliver the targets, but you have to embrace that slight uncertainty. But as Bill will explain, it's looking pretty good at the moment for these drugs. I should thank a lot of people here, past, not quite as many as Gregg, I think, but people past and present in my lab. The current group here, very many thanks to them, sunny day in Oxford. The funders, very important. The Wellcome Trust and Oxford University have been with me throughout this time. The Ludwig Cancer Research for the cancer work we now do. Importantly, at the moment, the European Research Council, very valuable message from Britain. Let's stay together. All these things are important. Three very important people I've mentioned: Chris Pugh, Patrick Maxwell, Christopher Schofield. And finally, my wife, Fiona. I've summarised 30 years' work in 30 minutes, so I just gave you the good bits. There have been bad bits to it, and Fiona has to suffer them, which she does very kindly. My family, and finally, my extended family, who arrived just a couple of days after the call from Thomas Perlman. And thank you very much for your attention.
Professor William Kaelin is the Sidney Farber Professor at Harvard Medical School and a member of the National Academy of Sciences. He is a medical doctor and cancer biologist. He earned his bachelor degree in mathematics and chemistry at Duke University in 1979 and remained there for his medical degree in 1982. He did his residency in internal medicine at Johns Hopkins University between 1984 and 1986, and thereafter in 1987 became a clinical fellow in oncology at the Dana-Farber Cancer Institute and Harvard Medical School. Dr. Kaelin was a staff and attending physician between 1989 and 2002 at the Brigham and Women's Hospital in Boston. And in 2002, he became a senior physician, Howard Hughes Medical Institute investigator, and professor of medicine at Harvard Medical School, positions he still holds. In 1987, at the time when his medical degree was completed and he had obtained his license as clinician, he joined the laboratory of David Livingstone, a renowned cancer researcher at the Dana-Farber Cancer Institute. While a postdoc in Dr. Livingstone's laboratory, he made several contributions on the mechanism of action of the tumor suppressor retinoblastoma gene product. In 1992, when he set up his own laboratory down the hall of Dr. Livingstone, Dr. Kaelin was looking for a project different from retinoblastoma, but he wanted to continue working on tumor suppressor genes. One such gene was the von Hippel-Lindau. Mutations in this tumor suppressor gene increase the risk for clear cell renal cancer, hemangioblastomas, and pheochromocytomas. And these are tumors that are highly rich in blood vessels, which was of interest to Dr. Kaelin. Among the many important scientific contributions on the von Hippel-Lindau protein, its role in the molecular pathway for oxygen sensing and clinical importance in cancer, Dr. Kaelin discovered that oxygen-dependent protein hydroxylation is the link between oxygen availability and adaptation of cells to low oxygen levels. I ask you all to join me in welcoming Professor Kaelin to the stage, where he will deliver his 2019 Nobel Prize lecture.
Well, as you can imagine, I'm both thrilled and grateful to be here. It is a little tough to go third. You've heard most of the scientific punchlines, and frankly, Peter is much funnier than I am, but I will do my best. And so for the opposite of funny, here's my financial disclosure slide, although the only one that's really relevant for today's talk is Fibrogen. So most importantly, here are the talented young people who've worked in my laboratory over the years, and they're really the heroes of the story I'm going to share with you today. This is only a partial list of the many generous and talented collaborators who have helped us on the work that's being recognized this week. I'd also like to thank my postdoctoral mentor, David Livingston. He's the one who taught me how to become a scientist. Frankly, before I met David, I was quite sure I was going to be a clinical doctor. And in fact, here I am as an intern at the Johns Hopkins Hospital as a medical house officer. And during this time, my chairman was the great Victor McCusick, and you've already heard Victor McCusick's name today. So it was really the great Victor McCusick who taught me to appreciate medical history, and he also taught me the power of human genetics. So I like to start this medical detective story where it actually began, which is with this paper in 1894 by Treacher Collins. So in hindsight, this is the first report of what we would now call von Hippel-Lindau disease. The English geneticist Treacher Collins had seen a brother and a sister, both of whom had an unusual retinal tumor, what we would now call a retinal hemangioblastoma. This is a blood vessel tumor of the eye. A similar, and I should point out that because these lesions are so vascular, it's easiest to see these lesions if you first inject the patient with a fluorescein dye before looking at their retinas. Now, a similar family was then identified and reported by the German ophthalmologist Eugen von Hippel, but I'm happy to report that it was really the Swedish neuropathologist Arvid Lindau who noted that these retinal hemangioblastomas were actually the marker for a systemic disease that actually affected multiple organs. And so to show you what some of these lesions look like, and on the upper left I'm showing you the direct fundoscopic view of a retinal hemangioblastoma, but as I've said, especially when these tumors are small, it's easier to see them if you first inject the patient with a fluorescein dye. But it was Lindau who noted that these retinal hemangioblastomas were often associated with hemangioblastomas of the cerebellum and spinal cord. So for example, here's a sagittal MRI of a patient with VHL disease, and this patient has a large cystic hemangioblastoma, which is seen enhancing here on MRI. Here's the solid component enhancing on MRI. These patients can also develop renal cyst and renal cell carcinomas. So here in the lower right, the urologist has unroofed a large renal cyst in a VHL patient and has discovered a renal cell carcinoma arising from the epithelial cells lining the cyst cavity. So with apologies to Treacher Collins, this came to be known as von Hippel-Lindau disease. It affects about 1 in 35,000 people worldwide. It's caused by loss of function germline mutations of the VHL tumor suppressor gene at chromosome 3p25. We've talked about the hemangioblastomas of the central nervous system and retina, and we talked about the clear cell renal cell carcinomas. These patients can develop adrenal gland tumors called pheochromocytomas, as well as a few other tumors. Now, I've highlighted clear cell renal cell carcinoma in red because hemangioblastomas and pheochromocytomas are fascinating but relatively rare. In contrast, clear cell renal cell carcinoma is by far the most common form of kidney cancer, and kidney cancer is one of the most common cancers in the developed world. Now, a word or two about the genetics of VHL disease. So here I'm showing you schematically the maternal and paternal copies of chromosome 3. Patients with VHL disease have inherited a defective version of the VHL gene from mom or dad. So in this example, it's from mom. But they're initially okay because they have one remaining wild-type allele. But what happens over time is a susceptible cell in the eye or the kidney will spontaneously lose or mutate the remaining wild-type allele, and that cell will then go on to form a tumor. So fortunately for me, shortly after I started my own laboratory, this paper appeared from the National Cancer Institute, where a group led by Marston Linehan, Bert Sabar, and Michael Lerman had actually isolated and cloned the VHL tumor suppressor. And they almost immediately showed what you would have predicted from the knowledge that germline VHL mutations predispose to renal cell carcinoma. Namely, if you now looked at non-hereditary clear cell renal carcinomas, you again saw that those tumors frequently had biallelic inactivation of the VHL gene, but here both hits, if you will, had occurred somatically, in contrast to VHL disease where the first hit had occurred in the germline. Now, the primary sequence of the VHL protein didn't really provide any clues as to how it might be involved in the control of cancer. But fortunately for me, my clinical training again turned out to be quite helpful. So, for example, I knew that the tumors seen in VHL disease were very rich in blood vessels or highly angiogenic. I'd already shown you on the left this characteristic appearance of a retinal hemangioblastoma on angiography. But I also know that I knew, for example, that kidney cancers were highly angiogenic. And in fact, in the pre-CAT scan era, the diagnostic procedure of choice in patients suspected of having a kidney cancer was to do a renal angiogram and to look for the characteristic appearance of new blood vessels in the kidney. Less appreciated is the fact that these tumors can also cause excess red blood cell production. And so, for example, normally if you take blood and spin it down in a centrifuge tube, about 40% of the volume will be made up of packed red blood cells, or in other words, the hematocrit will be about 40. But in patients with VHL-associated tumors, sometimes they make excess red blood cells and will have elevated hematocrits. And so as a clinician, I had studied lists like these. These are so-called differential diagnoses. In this case, it's the various conditions that are associated with excess red blood cell production or polycythemia. Now, in some of these causes, the increase in red blood cell production is clearly adaptive, such as in people who live at high altitude or people who have certain chronic lung or cardiac conditions. But in other of these conditions, this is clearly maladaptive. And it's known that, for example, some tumors are notorious for increasing red blood cell production through their ability to secrete either erythropoietin or erythropoietin-like substances. And it had always struck me as odd that the 3 tumors associated with VHL disease make this list, especially given the fact that cerebellar hemangioblastomas and pheochromocytomas are otherwise relatively rare. So summarizing, VHL-associated tumors are associated with increased angiogenesis, and that's because they frequently overproduce vascular endothelial growth factor, and they sometimes cause excess red blood cell production, and that's because they can produce erythropoietin. And what VEGF and erythropoietin have in common is that they're normally induced when cells or tissues are starved of oxygen. And so this suggested to me that there had to be some link between the VHL protein and oxygen sensing. So to begin to address that experimentally, we did northern blots for these 3 well-studied hypoxia-inducible mRNAs using cells that were grown under low oxygen conditions or high oxygen conditions. So for orientation, Hep3B cells are VHL proficient, and in these cells, as expected, the hypoxia-inducible mRNAs only accumulated under low oxygen conditions. In contrast, however, 7860 cells are a VHL null or defective renal cell carcinoma cell line, and in these cells, the hypoxia-inducible transcripts accumulated irrespective of oxygen availability. Othan Eliopoulos then corrected the VHL status in these cells by stable transfection. So here are 3 such subclones where he was able to restore the function of the VHL protein, and now he could restore the regulation of these hypoxia-inducible mRNAs. And this was specific because this was not seen when he stably transfected the cells with, for example, an empty expression vector. So to summarize, this said that a hallmark of cells lacking the VHL protein was that the production of hypoxia-inducible mRNAs was now uncoupled from actual oxygen availability. In other words, the VHL protein was strictly required for oxygen sensing. Now, in a parallel set of experiments, we showed biochemically that the VHL protein stably interacts with alongin C, alongin B, and Col2. And this turned out to be quite because my now colleague Steve Elledge had noted that alongin C looks like a yeast protein called SCIP1, and Col2, a member of the Kaelin family, looks like a yeast protein called CDC53. And at least in yeast, CDC53, when bound to SCIP1 and a so-called F-box protein, generates a so-called ubiquitin ligase complex that can target proteins for optimal degradation. So we began to suspect, likewise, this was a ubiquitin ligase. Moreover, when we solved the structure of this complex with the help of Nikola Pavletic, we saw that there were 2 hotspot regions for mutations in VHL disease. The first, the alpha domain, which we had already shown recruited the other members of the complex, and the second, the beta domain, which had the features of a potential substrate docking site. So then the question was, well, what was the substrate? Of course, we were aware of the work of Gregg, Peter, Frank Bunn, and others who taught us that many hypoxia-inducible mRNAs were under the control of the HIF transcription factor. And fortunately for all of us, Peter's group then published this landmark paper that he just described, where he showed that cells lacking the VHL protein were, for some reason, unable to degrade HIF-alpha under well-oxygenated conditions. And so seizing on this clue, we immediately showed that the VHL protein complex is indeed the ubiquitin ligase for HIF-alpha, and that, as we predicted, it actually does bind directly to HIF-alpha through the beta domain. So that's under well-oxygenated conditions. In contrast, when oxygen levels are low or the VHL protein has been mutated, such as typically occurs in kidney cancer, now HIF-alpha can accumulate, dimerize with its partner protein HIF-beta, and activate genes. So this was very satisfying. It explained why loss of VHL led to deregulation of hypoxia-inducible mRNAs, but it still begged the question, how does the VHL protein know, if you will, whether oxygen is or is not present, and hence whether it should destroy HIF-alpha? So one of the informative experiments in this regard is shown here. This is the work of Haifeng Yang in the lab. So here Haifeng was working with a cell line called TS20 cells. So these cells are VHL wild type, but they have a temperature-sensitive mutation in the E1 ubiquitin activating enzyme. And so as a result, when they're shifted to the restrictive temperature, polyubiquitination will stop, and proteins that otherwise would have been earmarked for proteasomal degradation can accumulate. So in the lower panel, Haifeng did an anti-HIF-1 alpha Western blot, and as predicted, he saw that when these cells were shifted to the restrictive temperature, HIF-1 alpha accumulated. But the truly informative experiment was the one he did in the upper blot, where here, instead of incubating the filter with an anti-HIF-1 alpha antibody, he incubated the filter with the recombinant VHL protein, and then detected the bound VHL with an anti-VHL antibody. And here he could see that the VHL protein could directly interact with HIF-1 alpha, which actually wasn't known at that time. But most importantly, it could only interact with the HIF-1 alpha that had accumulated if the cells were grown in an incubator that contained 21% oxygen. If the cells had been grown under hypoxic conditions, now a VHL could not recognize HIF-1 alpha. So this said something else that was important. This said that the signal had to be on HIF-1 alpha itself, as opposed to, for example, on VHL. Now, another informative experiment was this experiment done by Mircea Ivan in the lab. So Mircea mapped the minimal piece of HIF-1 alpha that could bind to VHL, and having done that, he could then synthesize that as a short 25-mer peptide with an N-terminal biotin that allowed then immobilize the peptide streptavidin and agarose for VHL binding studies. Now, at the core of this 25-mer was this sequence MLAPYIPM. So this peptide, when immobilized on streptavidin and agarose, could capture S35 labeled VHL protein, but only if it was pre-incubated with a mammalian cell extract to provide the, at least at that point, unknown activity. Now, fortunately, Caro at Thomas Jefferson had been doing linker scanning mutagenesis of HIF-1 alpha, and he had already shown that if you replace these 8 residues with 8 consecutive alanines in otherwise full-length HIF-1 alpha, that HIF-1 alpha mutant was now constitutively stable, and gratifyingly, the corresponding peptide could no longer bind to VHL. So next, Mircea did an alanine scan and determined that one of the critical residues was this proline residue. If the proline was replaced with alanine, now this peptide could no longer capture the S35 labeled VHL. And finally, knowing that the collagen Prolyl hydroxylases use oxygen as a co-substrate, and also knowing that iron chelators can mimic a hypoxic response, we hypothesized that maybe the modification was Prolyl hydroxylation. And this is an experiment now very analogous to the one Peter showed you a moment ago. When Mircea synthesized the peptide so it was already hydroxylated, this peptide could now capture VHL, and it no longer required a pre-incubation step, presumably because the relevant modification was already present. And Mircea went on by mass spectrometry and other means to confirm that the authentic modification in cells was Prolyl hydroxylation. And as you just heard, Peter's group reached the same conclusion independently and publishing contemporaneously. So with the help of Joan and Ron Conaway, Mircea Ivan then biochemically purified the HIF Prolyl hydroxylase. This Prolyl hydroxylase originally had the name Egelin-1. I will confess that as this work was being completed, we read the papers from Peter Ratcliffe's group and Stephen McKnight's group, where they had used clever genetic approaches in worms and fly, respectively, to get to the same answer. Suffice it to say, it's very clear. These are the HIF Prolyl hydroxylases. This is my schematic for how these enzymes work. Importantly, as you've already heard, they use molecular oxygen here to hydroxylate HIF-1 alpha. An important feature is that in contrast to the collagen prolyl hydroxylases, these enzymes have very low oxygen affinities, and hence they're sensitive to changes in oxygen over a physiologically relevant range. They require iron, which explains the effects of iron chelators. And finally, they require another co-substrate, which is variably called 2-oxyglutarate or alpha-ketoglutarate. Now, there are three members of the family, eglin-1, eglin-2, and eglin-3, with their alternative names shown in parentheses. But it's very clear that the workhorse or major regulator of HIF-1 alpha under normal conditions is eglin-1. And perhaps for that reason, you can't make it through mammalian embryogenesis, at least if you're a mouse, without eglin-1. So to circumvent this embryonic lethality, Andy Minamishima in the lab made mice where he could conditionally inactivate eglin-1 in adult mice using Cre-Lox technology and an inducible Cre. And when he inactivated eglin-1 in adult mice, he saw that the vessels got engorged and the paws got redder, and that's because these animals develop massive polycythemia. And as you've already heard today, we now know from the work of Joseph Cal and Frank Lee and others that likewise mutations in this pathway can cause familial polycythemia in man. So for example, some families will have homozygous or less commonly compound heterozygous hypomorphic VHL mutations, so-called Chuvash polycythemia, yet others have hypomorphic mutations in eglin-1, and yet others have hypermorphic mutations in HIF-2 alpha. And we also know that certain families or people who have adapted to life at high altitude will also have polymorphisms in this pathway. Now, in this model I showed you a moment ago, all of the EPO that's being overproduced in these mice is coming from their kidneys, and that's not surprising because the kidneys are the major source of EPO in adult mammals. But that's not true during fetal life. So during fetal life, it's your liver that makes most of the EPO, and then shortly after birth, the hepatic EPO locus gets silenced, and the renal EPO locus takes over. And this has both medical and economic implications because, for example, in the United States, there are over 20 million Americans with chronic renal failure, and at least 2 to 4 million of these are chronically anemic. So Andy Minamishima wanted to know whether the hepatic EPO locus could be reactivated in adult animals. And so to do that, he used genetic approaches in mice to inactivate the eglin family members either singly or in every pairwise combination, or he knocked out all three. And then he measured hepatic EPO mRNA levels in the black bars and circulating serum EPO in the open bars. And what he found was when he knocked out all three family members, he could see a robust induction of hepatic EPO, leading to an increase in serum EPO. And just for comparison, he also knocked out VHL itself in the livers. Now, about 20 years ago, I approached a biotech company called Fibrogen. They were making 2-oxyglutarate competitive inhibitors of the collagen prolyl hydroxylases. And the idea was that if we were lucky, perhaps some of their compounds would fortuitously inhibit the HIF prolyl hydroxylases. And if they did, perhaps that would jumpstart efforts to make specific inhibitors of the HIF prolyl hydroxylases. And so here, for example, is an early tool compound they provided to us called FG4497. When administered to cells in culture, it induced HIF-1 alpha, as shown in this Western blot experiment. But it was also active in animals. So, for example, Mikhail Safran in the lab made a mouse that ubiquitously expresses a HIF-1 alpha luciferase fusion protein. And when she gave this compound to these mice by oral gavage, she could see that HIF was stabilized as measured by an increase in light from these animals. So ultimately, this led to the lead compound from Fibrogen called roxadustat, and I've already declared my financial interest in Fibrogen, but several other companies are developing similar inhibitors. So here are data from the phase 2 trial in patients with chronic kidney failure. Again, this is an orally available inhibitor of the HIF prolyl hydroxylase. So here you can see that roxadustat induces endogenous EPO levels, and this is specific because here's the placebo in red. There's no evidence of pharmacological tolerance. You can come back 29 days later, and you can still see a similar response. So now you can basically turn on endogenous EPO at will. As you would hope, this translates into an increase in red blood cell production in patients who are anemic, here measured by looking at the serum hemoglobin levels, and you see a nice dose-dependent increase in circulating serum hemoglobin. At least for roxadustat, the phase 3 trials have now been completed and are starting to be published, and based on this clinical trial data, roxadustat has now been approved in China and Japan, and the US and Europe submissions will occur any day now. And this is just to remind you that in addition, if you look at least in preclinical models of diseases such as heart attack or stroke, these drugs also appear to be useful, but here much more clinical testing remains to be done. So now let's return to the role of VHL and HIF in kidney cancer. So once again, VHL disease has been highly informative. So I'll remind you that VHL patients are effectively VHL heterozygotes. Over time, they can develop hundreds of renal cysts, and when you look at these cysts, the cells lining these cyst cavities are now VHL null. So apparently VHL loss in the human kidney causes the development of preneoplastic renal cysts. And then over years to decades, some of these cysts will actually become true renal cell carcinomas. And when you sequence these tumors, you can see that there are almost always additional stereotypical mutations that represent cooperating genetic events. So one takeaway from this is that even though VHL loss might be a critical first step in kidney cancer, it's not sufficient to cause kidney cancer. Now, a similar picture appears if you study sporadic or non-hereditary clear cell renal carcinomas, such as has been done by Charlie Swanton. So what he and his group have done is taken renal tumors and done spatially distinct biopsies, including in some cases metastases, and then done deep sequencing and used the mutant allele frequencies to infer the evolutionary histories of those tumors. And almost invariably, he sees that loss of VHL is, again, the initiating or truncal event. But then he sees additional cooperating mutations that frequently occur subclonally or in a branch-like pattern, which gives rise to the idea of branch mutations versus truncal mutations. Which then, of course, begs the question, once you're a full-blown kidney cancer, is VHL function still relevant? Is HIF function still relevant? Now, frankly, Laureates in the lab years ago had shown that if you took a VHL null renal carcinoma cell line and restored the function of the VHL protein, these cells could still grow in a plastic dish, but they lost the ability to form tumors in nude mice. So then Keiichi Kondo in the lab took these cells and introduced into these cells a version of HIF, and particularly HIF2 alpha, that escapes recognition by the VHL protein because the Prolyl hydroxylation sites have been converted to alanine, and that restored the ability of these cells to form tumors. Conversely, he eliminated HIF2 alpha in various VHL null renal carcinoma cell lines. He used short hairpin RNA technology, but we now do this with CRISPR. And once again, you can see these cells will still muddle along on a plastic dish, but they lose the ability to form tumors. And so that said that inhibition of HIF, and in particular HIF2, is both necessary and sufficient for VHL to suppress the growth of renal tumors. And I should point out that, again, this is a specific property of HIF2 alpha. HIF1 alpha will not do this. Now, when we were doing this work, the classical teaching was proteins like HIF are not druggable. But fortunately, there were some druggable proteins that were controlled by HIF, and perhaps one of the most famous was VEGF. So by the 90s, a number of drug companies were developing inhibitors against VEGF, and we argued that these VEGF inhibitors should be tested against kidney cancer for the reasons I've shared with you today. In fact, on the next slide are data from Genentech, where on the y-axis they're measuring VEGF levels across a variety of tissues, and the tissue names are shown here. So in green are the VEGF levels for the normal tissues, and in the red dots are the VEGF levels in tumors derived from those tissues. So the first thing you see is that VEGF levels are modestly elevated across a variety of solid tumors, but that's what you would expect based on the knowledge that most solid tumors contain regions that are hypoxic. But clearly the 800-pound gorilla here is kidney cancer. It's in kidney cancer that you see the VEGF levels go through the roof, presumably because of this intimate relationship between VHL and HIF, and hence VEGF. Now, fortunately, a number of VEGF inhibitors have now been approved around the world for the treatment of kidney cancer. This has been a significant advance, and some patients do well for years on these drugs, but I should also hasten to point out they're not curative, and some patients don't respond at all. So we clearly have to do better. Now, I mentioned a moment ago that the classical teaching was that HIF was undruggable, but if you ignore that for the moment, I think you could argue just based on first principles that we would do much better to target HIF itself rather than any individual target gene. Now, fortunately, scientists who were then at UT Southwestern, Rick Bruick and Kevin Gardner, decided to ignore the existing dogma that HIF couldn't be drugged, and they identified a potentially druggable pocket in the HIF-2 alpha so-called PAS-B domain. And at UT Southwestern, they identified crude chemical scaffolds that could bind to this HIF-2 PAS-B domain, and in so doing induce an allosteric change in HIF-2 alpha such that it can no longer dimerize with ARNT and hence no longer bind to DNA. These were then out-licensed to a biotech company called Peloton that was recently acquired by Merck, and they did classical medicinal chemistry to make these scaffolds more potent, more specific, more bioavailable, and they were kind enough to share with us a tool compound called PT399 that was 1 or 2 atoms different from their lead. And in preclinical models, we showed that this HIF-2 inhibitor was highly active in preclinical models of kidney cancer, as did my former postdoc, Jim Bruguerolas, working independently. Here are data from the most advanced of the HIF-2 inhibitors. These are the phase 2 data for patients with advanced kidney cancer. These are so-called swimmers plots, where each horizontal bar represents an individual patient and how long they stayed on therapy once they were started on the HIF-2 alpha inhibitor. For orientation, this dotted line is 1 year on therapy. And again, I will emphasize these were heavily pretreated patients who had failed prior therapies, including VEGF inhibitors. And yet you can see that some of the patients clearly seem to benefit. So the yellow stars are patients who had a so-called resist partial response to the therapy. And the black arrows are patients who are continuing to do well with their tumors either stable or slowly shrinking. So this is highly encouraging, especially given how heavily pretreated these patients were. And this drug's about to go into phase 3 testing. So I started this talk in the late 19th century. I think it's time we move to 2019. So again, this is the first description in hindsight of von Hippel-Lindau disease, but we live in a very different time. So now the von Hippel-Lindau patients around the world are very well organized. They have a virtual community online, and this is helpful for a number of reasons, not the least of which is that they often know more about VHL disease than does their doctor, given how rare this disease is. So they can exchange information readily with one another. Now, importantly, Peloton also agreed to treat 50 patients with von Hippel-Lindau disease who had never been treated medically with a HIF-2 alpha inhibitor. These are patients who had so many individual tumors that they were in careful surveillance programs in an attempt to delay or avoid simply taking them to the operating room every year for yet another surgery. So there are these patients who have never been treated before, who have multiple measurable hemangioblastomas, small kidney tumors, etc. Now, the data from these 50 patients has not been publicly made available yet. It certainly hasn't undergone peer review, so that's my disclaimer here. But as another sign of the time we live in, these patients are posting on social media that they're responding to the drug. In fact, if you really look hard enough, you can actually find the pre and post scans that some of the patients are posting. I won't do that to you, but I will just point out, here's an example where a patient is saying, I never thought I'd see this day because their tumors are either shrinking or going away. And of course, this is very moving because these patients have had the sword of Damocles over their neck their entire lives. They've seen their parents and their grandparents suffer and die from this disease. So we're hopeful that maybe with the HIF-2 alpha inhibitor, we can start to change this. Finally, I want to recognize my late wife, Dr. Kaelin. Kaelin was a beloved breast cancer surgeon in Boston who survived breast cancer only to die of a glioblastoma in 2015. And she was my best friend and my soulmate and my partner in life, in addition to being the mother of my 2 beautiful children who are here, Kathleen Grace and Tripp. So I'd like to share this recognition with her. Thank you very much.
Dear guests, the 2019 Nobel Lectures in Physiology or Medicine have now come to an end. I would like to extend our gratitude to the Nobel Laureates for their extensive presentations this afternoon. Thank you all for attending, and thank you for sharing this significant moment with us. When you leave the hall, please leave the same way you entered. And now there's an opportunity to photograph the Nobel laureates on stage. May I ask the laureates onto the stage for a round of applause.